Welcome to the all viewers and well-wishers of the world. Purpose of my lecture is learn the knowledge from the word and return the knowledge to the word. Motto of my life is learn, earn and return. I have learned a lot, earned a lot. Now it's the time to return a lot to the word from where I have got all this knowledge. The topic of today's lecture is bacterial anatomy. That's very important. And bacterial anatomy means what bacteria contain inside and outside. Bacteria can live anywhere in our planet. All environment will grow bacterial species. Air contains most of the bacteria. By using TEM microscopy and SEM microscopy and gram staining, one can identify different species present in the each environment of the world. Unwashed hand contain more bacteria than washed hands. Parts of the bacteria can be divided for convenience into essential components and the non-essential components. First, we are going to talk about the essential components that contains nuclei with chromosomes, transposons, that is just like the nucleus of the other animal and the plant cell. Then cytoplasmic membrane, cell wall, outer membrane, teichoic acid, ribosome, mesosome, periplasmic space, cytoskeleton. And the non-essential components, these are the flagellum, pilus or fimbriae, spore, plasmids and the transposons, granules, capsules and glycocalyx. I will discuss everything in detail one by one, all these. So, first of all, the bacterial cell structure. If we just see from the inside to outside, the center of the bacteria that contains chromosome, and that is the nuclear region, which is called the nucleus because it is just like the nucleus. Then if we see there are certain ribosomes rounded structure inside the cytoplasm, these are the ribosome. Then some hollow structure like this, these are the plasmids. Then there is a constriction in the cytoplasm that is called mesosome. Pilus is a hair-like structure which project out from the cell wall, so that is the pilus. And then there is the outer membrane. Outside the cytoplasm, there is the cytoplasmic membrane, and outside of that is the cell wall, and then out of that is the outer membrane. So cytoplasm is bound by the cytoplasmic membrane, and then it is here one can see the cytoplasmic membrane. Then it is covered by the cell wall, and then out of that it is the cytoplasmic membrane. And out of all these there is one can appreciate the capsule. Now there is a space between inner cytoplasmic membrane and outer cytoplasmic membrane that is called periplasm. That will be also discussed when we will reach to that structure. Then there are certain polysomes. These are the food granules which can be used in case of the starvation or unhealthy weather for the bacteria. Then inclusion body, that is also a kind of storage granule which can be used in the starvation and harsh environment. So the structure of the bacterial cell, every bacterial cell have got main four parts if we just discuss it from inside to outside. Nuclei with chromosome etc. in the center. Out of that cytoplasm containing organelle, that is ribosome, granules, I have shown you in the previous diagram. Outer covering like cell wall and cell membrane, and structure external to cell wall, these are the capsule, flagella and the pili which are coming out from the cell wall, but these are lying outside to the cell wall. Now, if we just apply that from inside to outside, one can see this is the nucleus equivalent to the which is present in the bacteria as the nucleide. Then out it we see that is the plasmid inside the cytoplasm, ribosomes and depot substances that is metaphosphates, volatile glycogen granules, which are being used in case of harsh environment. Then one can see the cytoplasm is being bounded by the cytoplasmic membrane, and here one can see the thread-like structure which is coming out from the cell wall, that is the flagella, and here one can see that is the cell wall, and outside the cell wall this yellow line is the outer membrane. It is only present in the gram-negative bacteria, and these are the attachment fibers which are the pili or the fimbriae of the bacteria. So these are the parts. If we go from inside to outside up to the from the nucleide to the capsule, we can see the different structure which are the basic bacterial cell structure. Now coming to the nucleide, which is the most central part of the bacteria, that is also called bacterial nucleus, nucleus-like, so-called nucleide, so-called having the same function as the nucleus of the plant cells. This is the area of the cytoplasm in which cellular DNA bacterial chromosome is located in the form of nucleide. Single large circular chromosome, haploid in nature, attached to the cytoplasmic membrane, that is the septal mesosome. It replicates by simple fission instead of the mitosis. It has got 2000 genes. Human DNA has 100,000 genes. Advantage of 1N DNA over the 2N DNA is that the 1N DNA is more efficient and grow more quicker. It contains, the nucleate contains no nuclear membrane, no nucleolus, no mitotic spindle as in case of animal cell. So continued the nucleide, it has no histone proteins, no introns as in case of the eukaryotic DNA. So these are deficient, but polyamines and magnesium ions which work like eukaryotic histones. So these are present in the bacteria. Genetic material of the bacteria also consists of plasmids and the transport. These are also the pieces of the DNA, or these work like the DNA that contain DNA fibrils, but they lack a limiting membrane. Can be realized with the fusion DNA staining as oval or elongated body. Unfolded nuclear DNA is about 1 millimeter long. So here one can appreciate the central part of the cell, that is the nucleide, and also one can appreciate the flagellum, the capsule, the nuclear matter, cytoplasm. But remember the arrow showing the central part, which is the nucleus, and the other parts are also visible. The next part is the cytoplasm. It is the area below cell wall and bounded by the cell membrane. It has got two distinct areas, an amorphous matrix which contain ribosomes, mesosomes, nutrient storage granules, vacuoles, plasmid, and actin cytoskeleton. All these will be discussed one by one. The inner nuclear region, that is composed of DNA or the bacterial chromosome, that is the nuclear region. It is composed of 70 to 80% water. This is about cytoplasm. 20% salt proteins in the form of nucleic acid, carbohydrates, lipid, and inorganic ions that constitute about 20% of the total of cytoplasm weight. Stains with the basic dyes become granular with the age. It is the site for the cell biochemical activities which do occur most of the time in the cytoplasm. So one can see the cytoplasm, that is the area of, this is the DNA or nucleide, and this area is bounded by the cytoplasmic membrane. So whole this area which contain nucleus and the other thing, that is the cytoplasm. Here again one can see the space, and this is limited by the cytoplasmic membrane, and the structure which are inside, these are all the intracytoplasmic organelle or the structures. Cytoplasmic membrane is also called sometimes the plasma membrane. It is the limiting outer layer of the cytoplasm, or the boundary wall of the cytoplasm, barrier between interior and exterior of the cell, located just inside peptidoglycan layer of the cell wall, and composed of the phospholipids bilayer, as in the animal, chemically similar, but it is without the sterols. So the only bacteria, mycoplasma, which contains the sterols like the animal cells. This cytoplasmic membrane is flexible, weak, so ruptures very easily by the osmotic pressure of the nature. And here one can see the cytoplasm, and this is the cytoplasmic membrane, thin membrane that is being covered by the cell wall, which is brown and thick, and outside that is the capsule, which is of light color. Now, what are the functions of the cytoplasmic membrane? It is the active transport of the nutrients molecules which occurs through this membrane. So the things go inside the cell cytoplasm for the energy. Secretion of enzymes and the toxins out of the cell to save the cells from the drugs, that is also being produced by this cytoplasmic membrane. This is the place for the energy generation by the oxidative phosphorylation. So we can say these are the powerhouses of the bacteria, and it also have the role in the synthesis of the precursor of the cell wall, which is lying outside. So the raw material from here is being transported outside to make the cell wall. Now, the mesosome, it is a convoluted indentation in the cytoplasmic membrane. It is just like if you have a balloon and you put the finger in the inflated balloon, so the constriction that is going to appear, that will be just like the mesosome. It is a large tubular vesicular imagination of the irregular shape, more prominent in the gram-positive than in the gram-negative bacteria. That is the site of respiratory enzyme activity and the cell reproduction, and it synthesizes cross-wall septa, which helps the nuclear and the cytoplasmic membrane, cytoplasmic dwin during binary fission to make the cell into two parts. So that is the mode of reproduction of the bacteria. It is also a place of attachment for the bacterial chromosome, DNA, and that will become genetic material of each daughter cell. We do call the daughter cell, why not we call the son cell? I was unable to understand, because it is a man-dominated society, so perhaps it is why it is called the daughter cell. Here one can see this is the cytoplasmic membrane, and the arrow is showing the indentation which is just being produced inside the cytoplasm, and here one can see more easily that indentation that is being produced in the cytoplasm, and that is just the mesosome part of the bacteria.